thank you, organizers, for giving this opportunity to present my lecture on role of computer-aided drug design in modern drug discovery. So yeah, I'm not going to more detail into the CAD overview, and that I'm going to give more in case studies of CAD, and then both the structure-based and ligand-based drug design. So yeah, myself, I'm assistant professor for bioinformatics at BIOVIA State University. So yeah, we just been only 6 months here. So yeah, then this pandemic started, and then we are working from home. So yeah, I'm excited here at BIOVIA State University. So yeah, if you see the drug development process, I mean, yeah, Professor Tiwari gave a brief overview about computer-aided drug design. Yeah, I'm going to touch a few points on the overview of computer-aided drug designing myself, and then I'm going to go more in the case studies. So if you see drug discovery process, I mean, it takes 10 to 15 years, and then it has it looks simple: screening and then the lead identification and lead optimization, and then the preclinical development and clinical trials. It looks very simple in this page. So during every drug, where our computer-aided drug design is majorly involved, it's majorly involved in early drug discovery steps. That's where our computer-aided drug design, which makes the process, which makes process easy for experimentalists to test their molecules. So yeah, for drug discovery process, we need like extensive manpower. We need a lot of money, and then we need we need a lot of time. So we need a manpower, money, and time is needed for drug development process. So in order, what our CAD, I mean, computer-aided does is that it reduces the time, it reduces the, I mean, even the manpower for many experimenters, and it even reduces the money. So that's very important. But time and money is very important, especially in current scenarios. I mean, more important is the time, right? So we want to get as soon as possible a vaccine or drug. So especially in this times, I mean, this which makes the drug development process, even the vaccine process, even the most of the this computer, not exactly computer-aided drug design, but other computational techniques, which helps to make the drug discovery process simple. So yeah, in the preclinical development, where we do more, they do animal studies, and then in the clinical trials, yeah, they do on a testing of a drug on human volunteers. I mean, they will test the safety, efficacy, and the toxicity, and then if there are any side effects of the drug. So all this process is very lengthy. But in reality, if you see in the last slide, it's simple, but it's very complex. So yeah, it involves many scientists, right, from the biologists to chemists. I mean, even in there's a medicinal chemist, and even it is in the more on the pharmaceutical scientists, and even there are many the computational scientists now. Currently, even the CAD fellows involved in every drug discovery process. So in reality, it's very complex. If you see here, I mean, in each and every step, even in the target identification, right from the starting, the first step in a drug discovery process is a target identification. So if you see, there's very complex in the target identification. Where it involves data mining, and where it involves this understanding the hypothetical generation, and then the understanding the genotypes and phenotypes of our results. So it's even the target identification is very complex. And if you go for next steps, even if it looks simple, like talk about lead identification and optimization, but there are many people are involved in those steps. I mean, there are a lot of money and time is involved in each and every steps. So in reality, it's just very, very complex. So even if you see there are in a lead identity optimization, maybe this picture is not very clear. So it involves more in silico modeling, and then the medicinal chemistry is involved, and then there are process chemistries involved, and then we have to do safety, and then the prediction of the drugs, and then the pharmacologists are involved. So there are many, many people involved in the drug development process. So this CAD workflow is a basic workflow. Just now, Professor Tiwari gave a nice introduction about CAD workflow. So I'm not going to go detail into that. So majorly where it is involved, CAD, they are majorly involved in the two processes. One is a structure-based and ligand-based. Where do you use structure-based drug design, and where do you use ligand-based drug design? So if you know the structure, I mean, what do you mean the structure? Either it's a protein or enzyme. If you know the structure of the protein or enzyme, so you can go directly to structure-based drug design. Say, some case where you don't have a structure of a target. So in that case, you go for ligand-based drug design. So I'll tell you what are the limitations for a structure-based drug design and what are the limitations of ligand-based drug design. So in structure-based drug design, the first step is very important. If you know the structure of a protein, what do you do that? What do you do first step? First, you try to, if there is no active site, if there is no active site, what do? You try to predict a binding site or identify a binding site, means where the drug binds. So it's a very, very complex process. I mean, it's not that easy identifying a binding site. So we need to validate that. There are many binding sites in a structure. So you need to validate the binding site using both using literature and then the validation using experimental studies. So if you know the binding site, and then what you do is, if you know the structure and if you know the binding site, then you can test your molecules using docking and then the scoring. So this is the first steps in a structure-based drug designing. Whereas in ligand-based drug design, so where there is no structure available for a protein or enzyme, so then you go for ligand-based. Majorly there are two methods. I mean, mostly the pharmacophore modeling and then the quantitative structure activity relationship. These are widely used for ligand-based drug design. So we'll go into detail into the method in this CAD process in the next few slides. And then coming back to if you go for structure-based drug designing or whether you go for a ligand-based drug designing, you have to screen a lot of molecules. I mean, there are millions of molecules available in the many databases and stuff. So if you want to identify with novel molecules from the known molecules, you can go for a virtual screening. Both, either it is structure-based virtual screening or you can go for a ligand-based virtual screening. And then if you can identify the right, the top binding hits, so which are a strong binding to the protein, and then you go for lead optimization, and then you go for a new drug development. So yeah, this is all. Our CAD is involved more in the early drug discovery process. So yeah, this is very important because the companies, pharmaceutical companies, they don't want to waste money and time testing all the molecules they identified using experimental. So instead, they can go for a CAD process so that they can reduce time and money. So that's how it helps the early drug discovery process easy for drug development process. So next slide. Okay. First, there are two general types of CAD. That's why we discussed structure-based drug designing and ligand-based drug design. So in structure-based drug design, the first step is binding site identification. Means identify where means a hotspots or it's a binding pocket, which can fit the drug-sized molecules into the binding pocket. So what do you mean by hotspots? How do you differentiate between active and non-active binding sites? I mean, how do you differentiate? There are many methods to. One of the way to identify hotspots, I mean, where the binding sites where drug can be accommodated is we identify whether they have a hydrophobic surfaces, or we need to identify whether there are hydrogen bonding sites. Means these hydrophobic surfaces and hydrogen bonding sites, especially those sites, drive the ligand binding. So if you are testing a ligand, means on a protein or enzyme, so if your identification of these hotspots is very critical. So this binding site identification, if you see in the figure here, there are multiple potential binding sites. So yeah, we need to rank these binding sites and maybe which is active. Say if you are already known, there is already known ligands are there, then our job is easy. I mean, if it is already there is no much information about binding site, then you need to validate the binding site using these features. And then coming back to, if you know this in a structure-based drug design, if you know the binding site, then you do need to do docking and scoring. Is how the molecular docking is we all know, everyone knows, is one of the most well-known structure-based drug design. There are many types of docking. So now if you see, there are many softwares are available in the market, and then they use a different technology, different methodology. And if you see, before it used to be only blind docking, means a rigid docking. So especially like we talk about auto docking, it used to be more rigid docking. Nowadays, you can see there are various kinds of docking that makes your docking process more validated, like induced fit docking, and then there is a quantum mechanics polarized docking. So there are many which are more robust and more computationally intensive molecular docking methods are available in the market, and there are many softwares of both freeware and commercial softwares are available in the market. So I guess, I mean, tomorrow you're gonna do a hands-on session on BIOVIA software, BIOVIA software. I mean, maybe they explains more. That's how molecular docking can be done using different methods. So then, once you dock, means you have a 10-15 molecules. I mean, so when you're docking. So how do you score that? So we use scoring functions. We use scoring functions in scoring the drug molecules. So say telling about which is more top hit. I mean, which is the strongest binding. So means you need to score. We use the scoring functions. So many softwares use the different scoring functions for molecular docking. They use force field based, which is very famous. I mean, in the force field based, where you try to estimate the binding affinity, means we will even consider entropy, and then in the force field based. So this is force field based scoring function is more identified from the using information from the physical contacts. I mean, more in the atomic contacts. I mean, the drug binds to the protein. You know that there are physical atomic interactions. So use those information in order to estimate binding affinity. But whereas in an empirical, so empirical based, which is more based on the information about general which are now available market, like which is not very highly robust like a force field based scoring function. But whereas in knowledge based, so there you don't have much information about the interactions. So they use the statistical inferences from the general protein ligand interactions. So they don't have much information about the physical atomic contacts. So they just use knowledge based. These are the three very well-known scoring functions they use in molecular docking. So if you see here, there is a target. I mean, there is a ligand. Use a docking to identify the right binding conformation of this ligand. So you may use any other scoring functions. So in order to docking. So coming back to the ligand-based drug design. So you all know that when the QSAR and pharmacophore modeling is widely used for ligand-based drug design. So in the last case where we have a structure of a protein, so then it's easy to predict the protein ligand binding interactions. But if you don't have a structure, say if there is no crystal structure, means X-ray means crystal structure means either it is solved by X-ray crystallography or NMR spectroscopy, nowadays even electron microscopy. So if you don't have the structure of a target, so then what do? You can go for a ligand-based drug design. So this is because in the early stage of HIV drug discovery, they widely used ligand-based drug design. If you see the recently approved drugs, I mean, even the few decades, I mean, last decade approved drugs of HIV, they are more ligand-based drug design methods are widely used in early drug discovery process because there was no structures are available. Nowadays, because of there is a X-ray crystallography and NMR spectroscopy, or I mean, they developed a lot. I mean, nowadays they use robotic techniques to solve the structures. So before there was not much structures are available. So and then HIV was one of the best example you can say for how CAD can help the drug discovery process. And then they're widely used in HIV drug discovery. Nowadays, they use for many targets, many disease targets, is it from cancer or something? But the early days, it was very quietly useful, especially these QSAR and pharmacophore modeling were widely used. So in the QSAR, what is important? I mean, what is the first step in a QSAR? Why you want to go for QSAR? There are widely used QSAR methods are 2D QSAR and 3D QSAR. What do you mean by 2D QSAR? What do you mean by 3D QSAR? In 2D QSAR, where you use molecular descriptors, okay, where you don't have a structure of information of a ligand, so there way we can use only the molecular properties. So whereas in the 3D QSAR, where we use the structure of the ligand with the other physicochemical and properties. So what is the first step in a QSAR? So very important is identification of active ligands. So with the biological activity. So in order to do the QSAR, it's not only the just identifying ligands with the biological activity. You need to identify a congeneric ligands. Means there are diverse ligands which has a biological activity, and it means it should be a diverse structure, and it should be done in a same experimental condition. So that's very important. In a QSAR, you can't randomly select ligands from different experiments. So you can't select ligands. I mean, there are different assays available. There are many people. Say scientist X done using one assay, and they may identify a new ligands. Maybe scientist Y, he identified new ligands with different assays. So you can't use active ligands from different assays. You need to be the active ligands from same assay. So that is first major important is identification of active ligands with the biological activity. And then the second step is identifying the properties. So you got structures with the biological activity. Now what do you need to do? You need to identify and determine molecular descriptors. There are multiple descriptors available. It varies from software to software. So there are maybe 80, 90 descriptors are available. And the various, both the structural and physicochemical properties of the molecules will be identified. So identification of suitable descriptors is very important. And then once you know you have a ligands, you have a descriptors and the properties for all the ligands with the biological activity. So what you got to do is that you need to establish, mathematical expression relating these descriptors, which you determined with the biological activity of molecules. So it's very important. It has to be we need to discover correlations, right? If there is not proper correlation between the biological activity and then the physicochemical properties, then your model is not validated. So using that information, using the information of biological activity and then correlating with the molecular properties, you need to test those model, right? So yeah, you say that, no, I predicted a QSAR model, which is, yes, you have done a great, but whether it is really statistically stable, whether it is used for improving the properties of the ligands, whether this QSAR model is really helps in the identification of the new drugs or molecules or not. So that it's not only just identifying that you need to test this QSAR model with the statistical stability. I'll show you what I mean by statistical stability in my case studies. I'll show you what are the statistics we use for testing the stability of the QSAR model. We use both the correlation and cross correlation. I'll show you that in the next few slides. So in a QSAR, yeah, but whereas in a pharmacophore modeling, what do you do in a pharmacophore modeling? What do you mean by pharmacophore? So especially when you talk about in a compound, in a ligand compound, what do you mean by pharmacophores? Pharmacophores are nothing but features, especially which are hydrogen bond acceptor, hydrogen bond donor, where it's a hydrophobic moiety, or the positive or negatively charged groups. These are all pharmacophores in a molecule. So where we don't have a structure of a protein or a target, so then you go for say you have a known ligand, which you know that it shows some activity against some in experimental. So use those ligands, develop a pharmacophore, and you can use these features, pharmacophore features, you can use these features to screen databases in order to identify novel molecules. So if you see the pharmacophore here in the right hand side, it has features. It has a one hydrogen bond acceptor, which is in the green, and one hydrogen bond donor it has in the purple. So, and then it has two hydrophobic moieties in these molecules, and there is one positively ionizable charge group in this molecule. So you can, in ligand-based drug design, you can use QSAR modeling and then the pharmacophore modeling in order to identify the potent molecules for a particular target. So what are the advantages of this CAD? It is a cost-effective and the time-saving, and it's a rapid and automatic process. Yeah, obviously, as Professor Tiwari he explained about, there are limitations. Yes, there are limitations, but day by day, with the improvement of the softwares and with the improvement of hardware in the GPUs and clusters, it makes more validated, and more nowadays people are believing that CAD can be used as a tool in the drug development process. So we can minimize the synthetic and biological testing efforts, and we can obtain the drug receptor interaction pattern. These are advantages of CAD. In modern drug discovery, so with the advent of AI, right nowadays everyone talks about artificial intelligence. Artificial intelligence is there from many, many years. It's been from 1970s and 80s. They used artificial intelligence in a different way. But nowadays, there is a buzzword, the artificial intelligence everywhere, even in the automatic cars, and even in the energy technology, or in the, right now Tesla, and the advantage of automatic cars. So AI became very, very popular. It's not only the first wave of AI. They used before even 1980s and 90s, and then it came down. Again, they tried to use the AI, but nowadays, the real power of AI is in the market. I mean, even it exceeds a human technology, whatever it exceeds even human calculations and stuff. So with the advent of AI and hardware, it's the CAD which has emerged as an efficient means of identifying potential lead compounds, and then which will aid for the development of possible drugs for a wide range of diseases. It's not only for specific diseases they use CAD nowadays. With the advent of AI, they are and hardware, they are testing for multiple diseases, multiple targets. So especially the availability of supercomputers, and then the parallel processing, advanced softwares like BIOVIA and the other softwares, has greatly facilitated the rate of lead identification in pharmaceutical research. So artificial intelligence in drug discovery, so which is AI has surpassed human expert in terms of performance in many, many areas. If you see many areas, really we can say it surpassed, it replacing many humans in many jobs, many IT jobs, and many even in the grocery stores and stuff. It replaces humans. So it has surpassed human expert in terms of performance. So in many areas, there is no exception in drug discovery too. In drug discovery too, the artificial intelligence, and especially deep learning, we call about particularly deep learning approaches, which is what happens. This bringing, the deep learning, especially deep learning in past 5-10 years, it's really bringing changes in the drug discovery process. So AI is nothing but is being integrated in most aspects of early phase drug discovery, including target identification and lead discovery and lead optimization, and then pharmacokinetic property prediction, and then the toxicity prediction. Even in the clinical trial structuring, even AI is used. Nowadays, every area, in each and every area, AI is used. And very exciting is that in AI in drug discovery, if you see companies in US before 4 years, there were only very few companies, around 10 to 15 companies in AI in drug discovery. Nowadays, and in recent days, I've seen somewhere in one report, it shows that there are 250 companies in US which are AI based drug discovery. You can understand the fast pace of the AI and how it helps in the drug discovery process. So these are the drugs which are used. They use a CAD as one of the tool in the early drug discovery, especially if you see in the drugs like indinavir, ritonavir, and sacubitril. These are more the drugs which are targets HIV-1 protease, and you can see the raltegravir, which is approved in the recent years, which is in 2007, which targets HIV-1 integrase. I'm going to talk about it today. I'm going to talk about a lot about HIV-1 integrase. So yeah, I'm not going to talk about more on HIV-1 protease, and I'm going to talk about more on HIV-1 integrase. You see here, these are the list of the drugs which they used in CAD in the early drug development process. So this is not mostly updated one. There are many drugs are available in are in preclinical, I mean in the phase clinical trials, in the phase 2 and phase 3 clinical trials, so which is not listed here. And very one of the success stories of this CAD is a raltegravir we can talk about, and there are many other HIV drugs, because they use the CAD widely used in HIV drug discovery. So yeah, coming back to, yeah, that's all about overview. I'm going to give 2 case studies. One case study which is using combined ligand and structure-based approaches for identifying the HIV-1 integrase strand transfer inhibitors. So yeah, in the case study 2, I'm going to talk more on about structure-based visual screening and structure-based drug design. In the case study 1, which I've done mostly during my PhD and postdoctoral studies. So why we use combined ligand and structure-based? Why not just ligand-based? So there are limitations for structure-based design. That's what I've told. There are limitations for ligand-based drug design. Using those 2 techniques will help to validate those drugs as potent drugs, potent molecules. Potent molecules. So if there is no structure, yeah, we go for a ligand-based drug design. I'll talk about how I have used these combined ligand structure-based and ligand-based approaches in this study. You all know AIDS was first reported in 1981 and subsequently isolated in 1983. Around 38 million people globally were living with HIV at the end of 2019. And then you can see that it's been more than, it's been around 40 years, around 39 years, but still there are globally, it is affecting many people. So it infects vital cells in the human immune system, such as helper T cells, macrophages, and dendritic cells. It's a family of Retroviridae. I'm not going into more detail, and everyone knows about interaction. Yeah, coming back to this overview of replication cycle, starting from attachment to maturation. You see this, the red colored here. There are many stages of HIV life cycle. They try to use inhibitors to block that process. There are many stages, from the attachment to fusion, and then there is in a uncoating or a reverse transcription or integration, or in the assembly or in the release or maturation. There are many. There are 13 stages where they tried and tested identifying inhibitors for HIV-1. So today, I'm not going to talk about many stages of this schematic cycle. I'm going to talk about more in the stage 6, which is HIV-1 integrase, which is involved in the integration of the virus into the nucleus. I'm going to talk more about this integration process. So if you see here, I'm going to talk about the 2 steps. One is a strand transfer inhibition, and then the one I'm talking about viral host protein-protein interactions. In the first case study, I'm going to talk about strand transfer inhibition. So many integrase strand transfer inhibitors with the different chemical scaffolds targeting this strand transfer reaction have been developed. Especially recently approved drugs is raltegravir, elvitegravir, and dolutegravir. These are the 3 approved drugs for targeting integrase strand transfer inhibition. But still, they undergo drug resistance. That's a big problem, right? Especially when you're talking about viruses, the big problem about identifying drugs is drug resistance. And then the mutation. Mutations and drug resistance is one of the major problems. Even though there are many approved drugs for HIV, but still for many they are undergoing drug resistance, and then they're trying to mutate themselves. So in this study, in my study, I've used 128 integrase strand transfer inhibitors is collected from this literature. There's all these 128 molecules are dihydroxy isoindole derivatives. They are all reported from same research group and same assay. That's very important. Do you remember I'm talking about QSAR studies where it needs to be done with the same assay? So here have you used 128 molecules from which are developed from using the same assay. So on applying these scoring functions, what we do initially, we developed pharmacophores. So in order to identify diverse compounds with this pharmacophores, which is one of the best pharmacophore, which has this is AA means which is acceptor, D means donor, which is H means hydrophobic moiety, R means aromatic rings. So if you see here, this pharmacophore has 6 features, which is 2 acceptors, 1 donor, and then the 2 aromatic rings, and then 1 hydrophobic region. So what we have done is that we have plotted these molecules again as activity versus predicted activity from the QSAR model. If you see here in the training set, so we are divided into training set and test set so that we need to validate our models. So we have used the 75% molecules in the training set and then the 25% of molecules in a test set. And if you see then the training set on the left-hand side, which is plotted again is the actual activity, which is we collected from the literature with the experimental studies, and the predicted activity based on these pharmacophore models and QSAR. So we plotted, and then it was R squared, which is 0.8. Anything, that's what I was talking about, statistical analysis. That's what we were doing. You need to be more, if it is more closer to 1, which is more better the model is. So if you see the R squared, it may not be should not be overfitting, which is a 0.99. Sometimes it overfits the model. Even that is itself something is wrong. There is a noise in your study. So it is very good. I mean, which is 0.86, which is R squared for the training set, and the test set it's around 0.7. So here for test set, I mean, like for training set, we have used around 96 molecules, and test set we have used around 28 molecules, around 29 molecules we used in the test set. And if you see this both models with the scatter plots, which is well validated. I'll just show you the statistical results of the 3 pharmacophores which were developed, and then the QSAR results for the using that pharmacophore model we have used developed a QSAR model. So this is one of the best model is that which is AADHHR, which I've shown that in the which is highlighted in the red color. So which is this particular hypothesis. We use those hypotheses to develop a QSAR model. So here we are just a representation of the contours of a QSAR model. So what do you understand from this figure? Is that there are a lot of blue, and then there are red cubes, contours, and the blue contours. Blue contours is, and if you see in the A, which is developed for electron withdrawing property. So we are trying to identify using the QSAR model, which we have developed using the 128 molecules. So, and then the pharmacophore hypothesis. We are trying to understand whether these molecules, I mean, these predicted molecules, whether they are really helps in improving the model or not, whether it is a favorable region or not. For these contours, it shows that these blue contours shows that majorly these regions are which can be improved for electron withdrawing property. And in the B, where we can, there are very, very few blue cubes, which it shows that region where we can improve the property of hydrogen bond donor. And then on the C, we can see which are the favorable and these regions are very, very favorable for hydrophobic property. So these pictures, these contour picture representation shows that which are favorable and unfavorable regions for the QSAR model when the QSAR model is applied to this compound. So we see it shows that there are more favorable regions in this figure than the non-favorable regions. And what do you mean? Then we have developed a model. We developed a pharmacophore model, and we need to test it, right? So you can't blindly use that. You developed a model with the AA, which are 2 acceptors, 1 donor, 1 hydrophobic region, and 2 aromatic regions. You've developed a pharmacophore model, and then you have developed that, and you have even developed a QSAR model using that information. Then, whether you can use that model for visual screening or not. No, unless and until we need to test it more. I mean, you can't blindly use it. Just say this, your software given some model-based model. So you can't blindly use that model for a screening process. So we need to validate more. So how do you validate whether your generated pharmacophore model is really, which helps to identify novel molecules or not? Whether it is able to identify active molecules or not. So we need to validate it. So what we do is that we take that pharmacophore model. What do? We mix those active ligands. We collected active ligands around 237, and then the decoys means which are randomly, we take some decoys so that we see that whether our developed pharmacophore model is really is a good model or not. So we use that model. If you see in the figure here, in the x-axis there is a specificity, and then the y-axis we can see the sensitivity. Here it shows from 0 to 1 specificity, and then we mix both these molecules, and then we screen those molecules against this pharmacophore. If you see the plot here, by the 20% of screening reached, there were almost 70% of the molecules are active molecules. Here is a 0.7, means for better understanding, I put a 0 to 1. Means here, by the time the 20% of the screening is reached, there are 75% of the active molecules are there. Only 25% are decoys. Means your model is like which is a more better model, which is say, because why is a better model? Because it is able to distinguish between active ligands and decoys, right? Because the top hits, if you see the top hits of the screening is mostly active molecules. Means this model which you developed is in a pharmacophore model is more robust and more validated. Means it's able to identify more active ligands than decoys. And then if you see this is more looks like in the percentages. So if you see on the x-axis you see the percentage screen, and the y-axis you can see the percent actives found. So if you see here, that's what I was talking about. In that large figure is not very clear about in the percentages. Here, for your better understanding, I put in a percentage. So by the time 20% of screen is reached, there are more than 60% of actives are found. You can see that. Then this shows that the model which we developed is more validated. So it's very important, because it's computational. Sometimes you get a false positives, right? You know, we talked about the other professor, which he talked about. There are limitations. Yes, that's what the thing, because it gives false positives. So whenever you are doing any studies, we need to validate further using other statistical inferences and stuff. So this is one of the way we can validate your experiments. In the same way, even this is for ligand-based drug design. Even in the case of structure-based drug design, you need to validate further using many techniques. You need to using many binding free energy calculations and entropy, and you need to use many techniques for that. So this shows that the percentage screen versus percentage actives found, it shows that these model is wonderful for screening. Then I gave the numbers in order to show that. So we identify 148 out of 237 ligands, and only 20 of in number wise I given based on the numbers, and only 20 of 998. How come I gave top 20% of the rank? Hits. That's what I was telling about in the last. Both ligand based, so there was not use prototype. Let me know if I'm out of time. Please ping me. I didn't check the time. I don't know how much time I left.
No, you have sufficient time. You please continue.
Okay, yeah, just let me know, because sometimes I'm going out of. I'll try to stick to time. So just let me.
Okay.
I didn't check the time exactly. Okay, and then when we have done a docking using, and then we validated for that with the prototype formivirus, and that's how we have used both ligand-based and structure-based approaches. Then we superimposed the structures to understand the formivirus. So it's very, very important. In some cases, you don't have a clear picture of a target. It's very important to use both approaches, both ligand-based drug design and structure-based drug design. This is one of the best example I can show. We have done this, and then we have done docking, and then we have done simulated that, and then we try to identify whether these ligand is able to stabilize or not. So this is just a figure showing that that how these ligand is stable using MD simulation. So,
Dr. Reddy, please conclude within 10 minutes.
Okay.
Dr. Reddy, can you hear me?
Please? Yes, I can hear you. Thank you. That's fine. Yeah. So this is model which you have developed is enriched. We have validated using that. Then we have used the structure-based drug design, and then we have used MD simulations to further validate testing. We published this these studies in 2 journals. We have used this both the ligand-based and structure-based drug design studies. So that is one of the best study. Last was for ligand-based and structure-based drug design. Now I'm going to talk about case study 2, where we have used structure-based drug design. How we can use structure-based. Here we are talking about blocking the interaction between HIV-1 integrase and human LGF, so that you can easily understand. So it has the key residues in the binding interface for the integrase and LGF. These are the key residues at the interface. We try to use this information to identify user structure-based drug discovery. I'll tell you how we use this information for structure-based virtual screening. So fortunately, we have a crystal structure, which is with the HIV-1 integrase and then the human LGF. So we have seen the key contacts at the interface. What we do is that there are many contacts. We need to identify what are the key contacts, so that we can use that key contacts information for identifying drug molecules. So what we have done is that we try to develop a mutex, means alanine scanning mutagenesis, means mutating the each residue into one single mutants and double mutants. So you can't mutate everything at a time. So then it lost the protein function. So what we do, we try to do single mutants and double mutants and validate which are the critical residues, which are the hotspot residues, so that we can use those hotspot residues for virtual screening. Using those information, we have identified one of the critical two hotspots was a 168, which is a residue 168, and 170, which is a glutamine and glutamic acid, aspartic acid and glutamic acid, which I use this. You can see the very unstable in this system. Then what we do, we use that information, and when we go for with different databases, as Cambridge databases and other databases, we use that databases, and then we try to screen molecules, some thousands of molecules, using those hotspots which you identified from the metagenesis. So that we have used virtual screening studies, and we identified is a new novel molecules. This is one of the hot area, which is viral host protein-protein interactions, which I'm doing right now. So even I'm trying to develop this landscape for using AI and machine learning. I'm trying to do for all HIV viral protein interactions. Sorry, HIV and human protein-protein interactions. So yeah, it shows using our metagenesis, we identified two key residues. So it really binds to the 170 and 168, which especially this key residue of 170, our ligand binds. So that shows using this integrated approach, using mutational studies, MD simulations, and structure-based virtual screening, and MMGPSA binding free energy, we identified molecule that can block these protein-protein interactions. So it may not be very successful for all protein-protein interactions, but in this case, this is one of the good studies. We collaborate with the experimental studies, and they are doing this, continuing these studies further. So these computational methods we have used, and the hits identified in this study, which provides excellent guide to disrupt these HIV-1 integrase and human-led GF protein-protein interaction. So this is why we have published the work blocking this interaction. Now we are trying to collaborate with experimental scientists. And then I want to thank Bowie State University for providing nice facilities, and the BSc students and the former advisors. I collaborated with I've done my PhD in postdoctoral studies. We are located I mean, if you see the outer loop of Washington, D.C., it's just maybe 20 miles from Washington, D.C., which is a hot area. Which is we are rightly located into the areas where NIH institutes. There are 23 NIH institutes who are there within 50 miles of Bowie State University. Can you imagine? So I try to collaborate with many. Now I recently joined past 6 months of trying to collaborate with for experimental studies on this. So yeah, we are located in the prime location. So I'm trying to establish my lab. So